And today we're going to go ahead and talk with you about ways we can transform medical research, specifically through scientific and ethical innovation and advancements. And we're going to talk about science and ethics because these two issues really can't be separated. But before we dive in, I think it's important to answer a key question, and that is, why aren't Americans getting healthier? Or as some docs would put it to you, why are our children and adult patients getting sicker? So this question was posed to the former director of the NIH, Dr. Francis Collins, by an NPR reporter. And it's an important question because taxpayers invest heavily in NIH, FDA, CDC, and a range of other federal agencies that are tasked with advancing the health of the nation. One of the answers to this question is that we don't focus enough on prevention. The top killers of Americans today are heart disease and cancer, largely preventable diseases. 4 out of 10 cancers are now preventable in ways we understand. And when researchers look more deeply at actual causes of death, what they found is that dietary risk now surpasses tobacco use as the #1 actual cause of death, sometimes referred to as behavioral causes of death. And more deeply, there are social, economic, and environmental determinants of health that are determined further by legal priorities, political priorities. That's where you all come in. Really important, and social, economic, and environmental determinants of health are now so important to medicine that we have medical building codes for them. The other problem is that we don't invest in prevention research. Spending does not reflect research needs. And when NIH researchers have looked at this, what they found is that though 57% of deaths are attributable to 10 leading causes of death, only 34% of NIH projects focus in this area. And we see these disparities especially for dietary risk and for tobacco risk. So we don't invest enough in prevention, we don't invest enough in prevention research, and when it comes to non-clinical research, we don't invest enough in new approach methods as we do animal experimentation. There is a problematic reliance on animal experiments. Before we ever get to clinical research, there are massive investments in discovery and development and preclinical research, now called non-clinical research. The FDA Modernization Act, the passage of that over the holidays with the Omnibus bill, may change that, and that's good news because when you look at human bioavailability data and animal bioavailability data, it doesn't match up, it doesn't correlate well. So you should be able to draw a straight line through this graph, and you can see that the graph is all over the place. And when you look at dog data, this is true, represented in the red triangles. Primate data, this is true. And when you look at rodent data, this is true. Bioavailability just refers to the amount of an active substance in the bloodstream that can go and do the job that it was intended. So you all probably know this, but it's worth saying out loud that when we're making daily clinical decisions, we are not relying on animal experimentation. We use a hierarchy of evidence in medicine, and if they're available, we use systematic literature reviews and meta-analyses. That's filtered information that pulls together unfiltered information in the form of randomized controlled trials, cohort studies that either look forward or backwards at various risk factors or the effects of drugs or other interventions over time, and case control studies that look at cases and exposures that those cases have in common. This is how we know HPV causes cervical cancer. The other big question we ask is one of generalizability. Is the research population in the study similar enough to the patient or patient population that I'm treating? So for example, if I have a 65-year-old woman with heart disease sitting in front of me, I'm not going to pull a study that only focuses on men. Similarly, if I have a 75-kilogram man in front of me, I'm not going to pull a study that is just of mice with different sets of whiskers and a tail, right? And so this really begs the question of if animal research is sufficiently evidence-based to be a cornerstone of biomedical research. And when researchers and doctors have asked this question and used the hierarchy of evidence that we use in medicine, systematic literature reviews and meta-analyses, for example, what they have found is that generally no, it is not evidence-based to be a cornerstone of biomedical research. So why does animal experimentation fail? Well, we look at external factors and internal factors. And external factors relate to that question of generalizability that I asked. So there are disease-specific disparities. Other animals don't develop heart disease like humans do. Other animals don't develop certain cancers we do. Other animals don't have permanent breast tissue like humans do. And so there are a number of anatomical and physiological and other differences that matter. Individual health status also matters, and I'm going to come back to that. In animal experiments, the groups of animals are very homogeneous, sometimes from the same genetic lineage, for example. It's very different from the patient population that we're treating, or even who we want to enroll in clinical trials. There are also differences in outcome measures. How might we ask a dog if they have a headache, for example? Very common side effect reported with drugs in clinical trials and in patient populations. There are also epigenetic effects. Epigenetics is basically just how our environment influences the expression of various genes throughout the course of our lifetimes and ultimately disease risk and how disease progresses and how responsive we are to various interventions. And then finally, animal experimentation can't replicate the human genetic variability that we see in the US and across the globe. The other reason that animal experimentation fails is because of internal factors, internal threats to validity. So animal experimentation in terms of the way that research is conducted is quite different in terms of standards compared with human research. But the other issue more deeply is that we can't separate the experimental outcomes that we're interested in from the acute and cumulative trauma of laboratory life. We now know in humans, and we now know in non-human animals, that things like family separation, things like prolonged isolation and captivity ultimately affect the brain, the heart, the immune system, and every other organ system virtually in the body. So because of these and other problems, there's been a real move, especially in the field of toxicology over the past 10 years or so, what's called Tox 21. 21st century toxicology is meant to be evidence-based, non-linear, as we say, pulling information from a diversity of different sources. So, for example, exposure databases, looking at chemical exposures and the effects over time, in vitro methodologies or cell-based technologies, and then also using computer technology, bioinformatics, machine learning, etc., to predict human disease outcomes of interest, reproductive toxicity, developmental toxicity, and cancer risk, for example. And so this is primarily being used in chemical toxicity testing. It's being used more and more in pharmacology. More generally, in research, there's been a move to new approach methods, non-animal methods. And Jim's going to talk about organ on a chip, which is really fascinating technology. There's also bioengineered tissues, like epiderm, that's meant to mimic the outer layer of the skin. There's a system that mimics the human immune system, relying on cell-based data and computer information and processing, and we can represent human genetic variability and epigenetics and everything here, because this is used to ultimately determine or predict how a human population is going to respond to a vaccine or a drug, a biologic. And then finally, there are in silico methods, which basically is just computer methods, mathematical modeling, even AI, and machine learning. And because of interest in this area, as well as other new approach methods, this market is expected to grow globally to $1.65 billion in 2023, but that's still a small fraction of R&D. So we really need to also push with values, and that's what happened in human research. In human research, after years and years of egregious, unethical experiments, Congress mandated the creation of a commission that produced the Belmont Report, which focuses on key principles: respect for autonomy, beneficence or do good, non-maleficence, do no harm, key tenet in medicine and medical ethics, and justice, increasingly important in medicine. And Dr. Johnson, Dr. King, and I have talked about the need for a Belmont Report for animals using similar principles as a way forward. And that would be good not only for animals, but also for people, because what we know is that better ethics leads to better science. We've seen that in human medicine, we've seen that in human clinical research. Science has become much, much more rigorous, and we can see that in other areas of research. And I just end with one last thought, that it wasn't scientists and it wasn't even doctors who pushed for something like the Belmont Report. It was the public and Congress responded?
My name is Jim Corbett, and I'm the Chief Executive Officer at Emulate. We are a small life science company based up in the Boston area. We have approximately 110 employees. We're predominantly made up of biologists, pharmacologists, and engineers. But our origins actually started over at Harvard. Dr. Don Ingber's lab at Harvard, and actually specifically it's the Wyss Institute for Biologically Inspired Engineering. And about 10 years ago, Don submitted for a grant from DARPA. And if you know how DARPA funds, they like to take on big, challenging issues. And the grant that Don put forward was the drug development challenge that you see. Now, up the slide, you have a timeline of how a drug moves through the process to get to market. Starting on the left is the drug discovery stage, and it moves into preclinical development, and phase 1, 2, and 3 of the clinical trial process, and ideally to approval. And you can see from the time frame, is from the references below, it's anywhere from 9 to 14 years to get a drug to market. If you look at the bottom of the slide, it isolates the cost of each one of those phases of the drug development process. And it's very clear that today, it costs over $2+ billion to bring a therapy to market. But if you focus on that middle part of the slide, it says it's the attrition rate. And what that means, it's the failure rate. If you're lucky enough to move from a preclinical development program into the clinic, 9 times out of 10, you fail. And you can see most of the cost and time associated with that. So when Don was looking to do his proposal to DARPA, he said there's got to be a better way, and it has to be up front. There has to be a better way to predict what's going to happen in humans than the current models that exist today. Unfortunately, he was fine. If you go a little bit deeper on why the failures occur, it's really in these major categories. 40% are due to lack of clinical efficacy, meaning it didn't hit that clinical endpoint that it had set out to do, therefore it's not approved. 30% due to toxicity, another 10% because of poor pharmacokinetics, that's the body's ability to metabolize this drug in a safe way. And then there's another 10% for other reasons. So the 90% failure rate is still with us today, and it's a challenge. But if you just focus on, again, the top 3 technical reasons for failure, it really does come back to upstream models. And the state of the art today is just not predictive of what happens in the clinic. Whether you're using in vitro assays or you're utilizing spheroids or 2D cell culture or organoids or the current paradigm of utilizing animal models. And today, to get into a phase 1 clinical trial, you must do at least 2 species, historically, to get into that clinical trial. And yet, the failure rates for decade after decade are at 90%. So that led the team to figure out a new way. And Don and the team, again, about 10 years ago, said, how do we build a model that's predictive of what's going to happen in the human? And they came up with the organ on chip. And if you see it, it fits in your hand. It's about the size of a USB thumb drive. It's clear plastic. It's a polymer that's flexible by design. It has 2 microfluidic channels which are separated by a porous membrane. Because of the design of this chip, it allows the biologist to recreate that organ on the chip. So our liver chip, for instance, has 4 different cell types from the liver. Our brain chip utilizes 6 different cell types for the brain model. And because it has the microfluidic channels, you can flow media through it, like blood flowing through your body. Nutrients can arrive, or you can flow drugs. You can test compounds to see does it have effect on that organ. And that was the breakthrough that occurred, again, from Don Ingber's lab back in Harvard. So our hope is the future is going to change, and the past models that have consistently failed will go by the wayside, and that we'll have a future that will be a more integrated, human-focused and human biology focused. So we offer different organ models for customers to work with, but the most powerful part of this technology is really what you see in the outer ring. This is an open platform, so our customers can build their own models themselves on what's of their area of interest. You look at the lower right, you see the team out at USC has developed a colorectal cancer model. This group has access to diseased tissue, so they can create a colorectal cancer chip and test therapeutics against that chip. The team at Duke, you can see, did a kidney model. The team over at the Pasteur Institute did a Shigella infection model. And most recently, we had a lot of customers who are using our lung chip to study SARS-CoV-2 infection. The important part about that outer ring, as you can see, is almost all of them are academics. And they need the grants, the funding to continue to push this science along so the evolution can occur. I can tell you there's no chance that the team in the middle ring is going to be able to do that. Recently, we're fortunate, a publication just came out in the Nature portfolio, Communications in Medicine. This was a very large study that we conducted to see if our liver chip could predict drug-induced liver injury. We tested this over 870 emulate liver chips across a blinded set of 27 known hepatotoxic and non-toxic compounds. Now, the headline from the article was that the liver chip was able to correctly identify 87% of those drugs that caused drug-induced liver injury, and all of them had gone through 2 species of animal testing before moving into the clinic. So that was actually a result even better than we had thought. We also make the statement that we had 100% specificity, meaning we did not falsely say a drug was toxic when it was not, which was important. It's important to the research community. And finally, in working collaborations with an economist who studies research and productivity, research and development productivity, we were able to estimate that if there was routine use of the liver chip just for small molecule drug development, it could impact the R&D productivity by $3 billion. So it's quite significant. In addition to the economics and the performance, there's also a real patient impact. Of those 27 drugs, 11 of them caused 242 patient fatalities and required 10 patients to get liver transplants. And so you can see on the right-hand side of the slide where how did we do versus these drugs that cause fatalities, and the liver chip was able to detect all 11. So again, I think a big win for the patients. Now, one of the criticisms we have is, well, you look at the date on those drugs and when they were on the market and pulled off, and certainly we've gotten much better than what you see in the slide there. Well, unfortunately, times haven't changed. And if you just look in 2022, the companies and the programs you see up here, these clinical trials were either stopped completely or halted and paused. And the issue of drug-induced liver injury is by far not behind us. It will continue to plague the industry until an adoption change starts to occur. So, in conclusion, I just want to give you a few more thoughts. As I demonstrate here, I think organ-on-chip technology has demonstrated superior performance to animal models, but more work is required. That was just one model from one company. Funding for the academic community and government agencies will help advance the science and allow for robust validation. That takes quite a bit. I think the FDA will need scientific evidence and validation to advance policies transitioning away from animal models. Luckily for us, they protect the consumers, and we're all consumers of health, but they'll need the scientific evidence to move away from it. And then finally, reducing unnecessary animal testing while increasing patient safety and lowering healthcare costs should be a unified goal for all parties.
I'm going to discuss a little bit of the history of research using animals and how ethical principles and regulatory guidelines for using animals in research should be aligned with the principles and guidelines for research with vulnerable humans, as described in the Belmont Report. So human and animal research share a common goal, which is to benefit humans by advancing health. And despite this shared aim, there are vastly different ethical and regulatory schemes which result in huge differences in how the research subjects are treated. So for humans, we have the Common Rule, which has as its underpinning the ethical principles outlined in the Belmont Report. For animals, there's the Animal Welfare Act and some specific guidelines that were established by various agencies that use and fund animals in research. Under the AWA, many animals that are used in research are exempted from regulation, including mice, rats, and fish, who are among the animals used most frequently. Rather than protect animals, the AWA protects industry and researchers. In the mid-20th century, revelations about research atrocities during World War II led to the worldwide promulgation of research ethics codes. But well into the 1970s, appalling research on vulnerable humans occurred in the U.S. Incarcerated persons, institutionalized adults and children, and famously in the Tuskegee syphilis experiment, persons who were vulnerable to manipulation and deception due to race and poverty and lack of education. In response to public outcry when Tuskegee was exposed, the National Commission for the Protection of Human Subjects was established in '76 and they wrote the Belmont Report. Belmont established principles for the ethical conduct of research that directly responded to the ethical shortcomings of research with human subjects at the time. So respect for persons, which is an obligation to respect autonomy and protect the vulnerable; beneficence, which is an obligation to do no harm and to minimize the harms of research; and justice, which requires fairness in the distribution of the burdens and benefits of research. Belmont's principles can be directly applied to research practices, and they work together. So respect for persons requires informed consent and requires that participation in research be voluntary and protection of the vulnerable is there. Beneficence requires that we justify research by thoroughly assessing the risks and the benefits. So brutal and inhumane treatment of human subjects can never be justified, and the risks must be reduced to just those that are necessary to achieve the objectives of the research. And you'll note here the connection between justice and justification, because justice requires the fair selection of research subjects, which means their use must be scientifically justified. And again, this is in response to the exploitation of vulnerable persons. Institutionalized children, for example, who were used in research that was not specific to children, which benefited others. It requires that the selection of subjects be scientifically appropriate. We cannot select vulnerable human subjects simply because it's easier to use them. So this was a bold and transformative idea that even if using particular humans could be beneficial for others, their use can still be unethical and morally prohibited. The benefits to others cannot justify using vulnerable humans who are easily coerced or manipulated, and those who are unable to consent or volunteer for research. These transformative changes in protections for human subjects had spillover effects for animals. And animals have borne much of the burden of protecting human subjects. So Belmont and other research codes prompted this scientific pivot away from using humans and onto using animals. But the same kinds and levels of protection do not exist for animals. And as a consequence, the number and diversity of animals used has increased significantly in the last 75 years. So the Animal Welfare Act was passed in response to the kidnapping of a Pennsylvania family's dog, a Dalmatian named Pepper, who was then sold to a lab and killed. And this is why so much of that law actually concerns transportation and the acquisition of animals. It began as an anti-dog napping law, and it was passed because of overwhelming public outcry about it. So the AWA, as it currently exists, protects the animal research enterprise, and it shields perpetrators of cruel and inhumane research who can simply say that they are AWA compliant. There are no protections at all for most of the animals used in research and only minimal regulations for others. And this is very much out of step with current scientific knowledge and understanding about animals and animal well-being. Compliance with AWA is barely enforced, and the welfare of many animals is considerably worse today than it was in 1966 when that law was passed, and worse across several domains, including in research. So the status quo is not working for animals or humans. Lax regulations for animal research, coupled with stronger regulations for human research, have led to the over-reliance on animals because it's easy and convenient to use them. You can place an order online and have your genetically modified mice delivered to your lab. At the same time, research on better human-relevant, non-animal methods is underfunded and stifled because the incentive structure favors using animals. But as we've discussed, animal research rarely benefits humans. More than 90% of therapies tested on animals fail to translate to humans. And in some areas of research, like Alzheimer's and stroke, two diseases that cause tremendous suffering and death, the failure rate is closer to 99%. So the failure of animal research also harms humans. humans in whom drugs get tested, who suffer serious adverse effects, and patients who continue to wait for effective and safe treatments. Belmont's principles are the right principles to govern human research, and comparable principles ought to govern animal research, because animals are vulnerable to the same kinds of harms that humans are vulnerable to. Yet animals are subjected to harms that are forbidden by Belmont and the Common Rule, including lethal research, captivity, forced breeding, amputations, and more. And this has been the legacy of protecting human research subjects. We transferred the harms to animals, but not the benefits. So for the sake of brevity, I will just focus on one of Belmont's principles, justice. Justice requires the fair distribution of benefits and burdens, but the supposed benefits of research are almost entirely on the human side, while the burdens are all on the animals. And those human benefits rarely materialize, while the burdens for animals are very, very real. The death, the pain, the distress, and the captivity. As Belmont conceives it, justice is not simply a lofty moral or political ideal. It's a scientific principle as well. Scientific justification is necessary for research to be just. And the use of animals in research, in spite of their differences and their demonstrated unsuitability as models for humans, is motivated by the inability to use humans. And that's a justification based on convenience and ease of use. It's not a scientific justification. Using animals in place of humans in science and medicine is ancient technology, thousands of years old, and it predates modern science. And it accelerated when human rights and the promotion of ethical research forced scientists to pivot away from using vulnerable humans. This was the easy way, the ancient way of resolving moral conflicts in scientific experimentation. But after thousands of years, it's clear it doesn't work scientifically, and it doesn't get us off the hook ethically. Only in the last century did we reimagine and revolutionize the ethics of research with humans, and it changed science. It's time to revolutionize science again and to reimagine biomedical science in a way that is fully justified as good and ethical science that promotes human health and protects humans and animals from harm. So Congress has a crucial role in modernizing and transforming science by promoting accountability and transparency, and by designating preferential funding to incentivize innovative and ethical science that advances human and animal health, and by pushing federal agencies to create roadmaps towards more ethical, human, and patient-centered transformative scientific methods.
I am delighted to have the chance to address you this afternoon as the last speaker in our panel. And as you can see, I am going to talk about a patient's perspective on transforming medical research. I'm a patient, but I'm also an anthropologist, so these two perspectives will come together. I taught for 28 years at William & Mary in Virginia, a college that may be known to some of you. I'm also an author of 7 books on animals and a freelance science writer. So I'm going to talk a little bit about what it was like to be a patient. Specifically, I was diagnosed 9 years ago with a particularly nasty form of cancer, uterine cancer, that required surgery, chemotherapy, and radiation. And following that, I had another life-threatening episode of a deep vein thrombosis, which is a massive blood clot that broke off and went to my lungs for a pulmonary embolism. This has led to a number of chronic issues, which I'm not going to belabor because it would take too long, but I can mention a couple. I have chemo-induced neuropathy in my feet. I suffered pelvic fractures, partially from the radiation, and my digestive system will never be the same after all of this. But what is important for my purposes today is to point out that I was a very fortunate patient. I had rights. Everything that was done to me basically was done with my consent. It was done with enormous skill and comfort for my benefit. And I was surrounded by people who wanted to make those side effects better. It was a difficult process, but one of the long-lasting effects of going through this has been that my empathy for laboratory animals has just soared exponentially. Because the animals that you've heard about today, the animals whose lives are used in medicine, whose lives are sacrificed not to the benefit of those of us who are patients, they don't have rights, they don't have comfort. What is done to them is not done to them for their benefit, and they don't have people, for the most part, easing their side effects. And what I think I can do today in this talk is echo what my colleagues have said in a lot of ways about what's needed in terms of transformation to bring together science, ethics, and justice, most certainly for the future, but to emphasize something about the incredible stress that these animals go through and why that's a harm for people like me, for patients and our families. And one key point that I really want to make is that all of us, no matter our age or our position in life, will be patients at one time in our lives. So although I'm talking to you as someone who's actively a patient, I do believe that this affects every single person and our families. So when I fight for animals in laboratories, and I do spend time doing this as an animal advocate, I feel very strongly, I know that I'm fighting also for patients, for so many other people, because we need a system that benefits us, a system that's ethics and justice centered that we do not have at present. So I'm going to take the liberty of rewriting a headline for you that appeared in Science magazine in December. The headline claims that the indictment of monkey importers could disrupt U.S. research. And what this refers to is the fact that many wild-caught monkeys are brought into this country, taken from the wild and put into these laboratories and experimented on with, as we see, a very high failure rate. I'm suggesting instead that reliance on animal models holds back U.S. research. And I think you've heard enough so far today to know why I would say that. Part of the reason I say that also, in addition to what you've heard about the statistics and the failure of validity and the failure of translation, is that I myself work in an adjacent space in anthropology. I work on animal emotions, and I understand how extremely altered are the lives of these animals in laboratories, emotionally and physiologically. At the very same time, they're being used as models to help patients. And something doesn't add up about this picture. So to talk a little bit about my work in animal emotions, I have documented for the last 8 years now, 10 years, that animals feel love and grief. These emotions don't belong to us humans. And it is not, as it is sometimes claimed, misguided anthropomorphism. It's actually very rigorous science to say this, to speak of joy and fear and love and grief felt by animals in the wild, in our homes, in zoos, on farms, and most contextually important for today, in laboratories. So many of you may know the story of Tahlequah orca, who you see here in the large slide. In the summer of July 2018, she gave birth in the Salish Sea in the Pacific Northwest, but her daughter died within an hour of being born. And what happened over the next 17 days was an expression of what scientists, including me, believe is love and grief. Tahlequah, the mother, did not let the small body fall off her own body into the ocean. She swam with it on her body. If it fell off, she dove and retrieved it, and she kept going, radically altering her own agenda, her own typical daily routine. People know about this orca. What people don't tend to know is how widespread the expression of grief and love is among animals. I have a picture just here of a donkey named Lena who had bonded in an animal sanctuary with a horse. And the horse had to be euthanized in old age. And Lena is standing on his grave. You see that here. She wouldn't come in, she would eat, she would sleep. She did eventually recover, but she felt this deeply. And the animals that are used in laboratories are just as emotional, just as sentient, just as an ability to feel, and just terribly altered in their normal lives. So I'd like to try to bring home not just animals in laboratories in the abstract, but a little bit about individuals and then the nature of how their lives are altered, which of course is my main point, that as a patient, I do not benefit from what happens to these animals, and I'd like to show you why. First of all, rhesus macaques are the species I'm going to focus on with my examples. They're a very common laboratory monkey. In the wild, they live in what are called matrilides, groups of related females that stay together their whole lives. They rest and groom and eat together, and they're very social. This is the same as the baboons that I studied in Kenya for my doctoral research of different species. Of course, you bring a rhesus macaque into a laboratory, and it's very, very different. Here in Wisconsin National Primate Lab is Cornelius, rhesus macaque, who's been held there since his birth in 2010. If you can think about the number of years since 2010, and think about the months, the weeks, and the days that he has endured this captivity, he's held in a very sterile cage, as you see. Mostly during his life, he's been alone. Occasionally, he's been with other monkeys. Even when he's with other monkeys, he never feels the sun on his face or fresh air, and he has shown extreme stress as a result of this captivity, extreme diarrhea, very clear depression, and other physical problems because he was used so invasively. I'm going to talk also about a second rhesus macaque, born in a different laboratory but also held for most of her life in Wisconsin. She was in fact killed in Zika research in December 2021. As you know, Zika is a mosquito-borne disease that harms humans. It particularly harms pregnant people, and it can cause birth defects. I'd like you to look really closely at this slide. This is the most important slide I'm going to show you. You see that Princess is also held in a sterile cage, and you can imagine when you compare that to the picture you saw of them in the wild with their matrilides, how much this restricts and changes and stresses her life. But the two things I want you to look for specifically are, one, there's an infant she has on her belly. 5 times over the course of Princess's life, she had a 1-year-old infant forcibly removed from her belly by laboratory workers. This would never happen in the wild, and she was depressed and stressed each time. If you also look at her body, I think you will see that this is an extreme case of self-mutilation from stress. Look at her body, there's no hair. Look at her body, it's raw. She did this to herself because of the conditions under which she lived. And I want to ask you 2 questions. Does your empathy emerge looking at her? And secondly, do you think this is a reasonable animal model to study diseases to help patients and help their families? Do you think this model on such an incredibly physiologically and emotionally altered animal makes sense? I think of Cornelius every day. I think of Princess every day, too, who is no longer with us. And now, our job is to scale up again and to think about the other 2,000 monkeys held at this laboratory, the other monkeys held across the world, and the dogs and cats and rabbits and ferrets and fish and octopus and rabbits, I could go on, who also feel their lives. So, as we've heard, the system is not working. We've talked a lot about how to make change, and I think one thing that's useful is to think like an anthropologist. This is a culture that's entrenched of animal models because the funding has been in animal models. And we see that that's changing. Well, what Jim talked about earlier, he used the phrase, I think this is a win for patients when we talk about organs on a chip. And that just lights me up. I mean, that can change our lives, all of us who are in these situations of chronic disease and our families. This is what we need to work for. So again, when I look at Cornelius and I look at Princess, and I am working on a court case involving them at this moment, I know I'm doing it for them, but I'm doing it for all persons who are patients. So we need to change this culture of funding. Funding, you'll hear it as people who are involved in Congress hear it all the time, and that's what we want to say to you. Preferential funding for advanced science that's not based on these miserably stressed animals. To recognize that we have to reward young researchers in universities and biomedical centers, and to work with taxpayers. Taxpayers don't want to fund these kinds of stressed animals that don't help their families. So the future is justice, science, and ethics all together, and the future is no more Cornelius and no more princess.